afternoon and welcome back. Today we're going to talk about unusual infectious agents. This is a lecture that the players don't really fit anywhere else in this course, so they get their own lecture. But I haven't given this in 3 years because of snow days in past years that one of them had to go, and this was it. But I like this a lot. I think you'll like it too. And this lecture really addresses this fundamental question, which is, what is the minimum genome size needed for an infectious agent? And could an infectious agent exist without any genome? Is that possible? Well, yes, the answer is yes. And we will see answers to these two questions from viroids, satellites, and prions. Viroids here on the left, satellites in the middle, and prions on the right, which are nothing more than infectious proteins. How about that? Let's start with viroids. These are nothing more than circular single-stranded RNAs. Probably what existed on Earth before cells in the RNA world. Just little bits of replicating RNAs, selfish replicators, if you will. And we think these are actually remnants of those days. So here's one illustrated. It actually is extensively base paired with itself, so it's not technically circular, but if you denatured it, it would be circular. But the other cool thing, besides it's just a piece of RNA, there are no protein coding regions. These entities, viroids, they have no protective coat, yet they can go from host to host, as you will see. And there are no cell receptors needed for infection. Now, I would say these are probably not viruses. I've been kind of waffling about this for a while, whether this is a virus or not. And I had a conversation with Eugene Koonin earlier this year in February, and he convinced me that they're not viruses because a virus has to have a capsid. That's what we think. Oh, that's our definition anyway. So these are not viruses. That's why they're viroids. And these get into plants. They replicate, and there are two families, Viroidae, which stands for potato spindle viroid. These replicate in the nucleus, and Viroidae, avocado sunblotch viroid. These replicate in chloroplasts. So that's how these are classified into two families. They are, as I said, circular single-stranded RNAs between 246 and 401 nucleotides. The RNA displays extensive internal base pairing and looks like a rod in the electron microscope. And some of these, as you'll see, are ribozymes. A ribozyme is an RNA that has enzymatic activity and, in fact, can cleave itself. And it's another remnant of the RNA world where RNAs had enzymatic activity. There were no proteins. And this ribozyme is essential for replication of the viroids that have ribozymes. And here I want to point out how these viroids are distinct from viruses in the way they reproduce. Viruses, of course, are parasites of the host translation machinery. They make mRNA. Their enzymes produce mRNA, and that's translated by the host cell. But viroids are parasites of the transcription and translation machinery, of course. So you go further back in the pattern of gene expression. They have to be transcribed and then translated, as you'll see. So the first one discovered, the first viroid, potato spindle tuber viroid, discovered in 1967. It was discovered during the production of potato seed stalks that was found to contaminate them. And this has been the prototype for the smallest known nucleic acid-based infectious agents. So without coding any protein, they can be infectious. This one's 359 nucleotides long. And depending on the viroid, some of them do nothing in plants, and others cause economically important diseases of crop plants. And so for farmers, this is a big issue. Here's one infecting a tomato plant, and you can see different levels of symptoms from healthy plants all the way through mild, intermediate, and serious symptoms. I think these are different variants of viroids that cause different levels of symptoms. And I can't resist to tell you my favorite viroids. I have 3 here, but they're many because they have just the coolest names. Kadang, coconut viroid. Causes a lethal disease of coconut palms, as you can see here. This very sad-looking coconut palm tree. Makes pina colada drinkers sad, of course, because it hurts the coconuts. Then we have hop latent viroid. So you can see they're named after what they infect, although sometimes their host range is broader than what they're named after. This makes no symptoms in the hop plant, which of course is used to make beer. So beer lovers are relieved. And then we have apple scar skin viroid, which causes mild symptoms in the apple. You can see it gives it this mottled look, and you may have seen apples in the market like this. They taste fine. There's nothing wrong with them. They're not soft or anything. It's just the skin, as the name would indicate. And people may not want to buy them, so you may not see them in the big supermarket chains, but maybe in local ones you might see them. But I would eat it. It's fine. There's nothing wrong with it. Anyway, those are 3 of my favorites. These are transmitted both vertically and horizontally via pollen. So here's an example of vertical transmission. Again, within pollen, the pollen has the viroid in it. Here it's attaching to the stigma of the plant, right? And it would be brought in by the pollen tube and moved to the ovary. And then you would have production of a seed, which would sprout into a seedling. But then the viroid is in the pollen, it gets into the ovary, it gets into the seed, and then the new plant has it. So that's vertical transmission. But then we also have horizontal via pollen again. Here we have a viroid-infected plant. It's shedding pollen, and the pollen is reaching the stigma of a second plant, makes its way into the plant. It could get into the ovule and end up in the seed again, or it can get into the cells of the plant, as this other arrow shows here, and infect the rest of the plant. And so obviously this pollen can be transmitted by bees, by farmers brushing against one plant and bringing it to another, by farm equipment and so forth. And that's how these are spread, mechanically, right? Not by going into a vector of some sort and replicating in that, because he's only replicating the plants. And a study of viroid RNA has shown that they have functional regions, as you can see marked here. The POSPI viroidae have left and right ends. They have a central conserved region, then regions that are important for pathogenesis, for causing disease symptoms in plants, and then there's a variable region as well. And this is on the bottom, peach latent mosaic viroid, an avsun viroidae. This is more extensively folded viroid. It's longer. And this one has a ribozyme. So these shaded areas are parts of the ribozyme, and they will cleave themselves. They cleave themselves, and that happens in replication, as you will see. So how do these replicate? Well, they're amazingly, they're replicated by host RNA polymerase II. They make concatemers, which are one molecule after another. We saw that with herpes simplex DNA replication, but they're copied by host polymerase II. This is the only example, as you'll see, one other we'll talk about today, where these RNAs, these viroid RNAs, are copied by host Pol II. And one group, of course, forms this hammerhead ribozyme. That's the name of it. And a ribozyme is an autocatalytic self-cleaving RNA. This ability of RNA to cleave itself was discovered in 1981. You don't need protein for this self-cleavage. That got a Nobel Prize. And this, as you will see, is used to cleave multimeric structures that are produced during replication. There are viroids that don't have ribozymes, and they're cleaved, their concatemers are cleaved by nuclear enzymes of the host. So how do these replicate? Here's an example for members of the POSPI viroidae. After they get into the cell, the circular viroid RNA gets into the nucleus. It is copied by Pol II, which is amazing that it works, but it does. And you get concatemeric RNA. So the red is the complement there. And there's 2 whole genome lengths. That's what we mean by concatemer, but they can be longer. These get imported into the nucleolus, and there they get processed by cell enzymes to make genome length RNAs. They circularize, and then they get exported out of the nucleolus, and then they get out of the nucleus, and then they move to the next cell through these plasmodesmata, which connect the cells of plants. On the right is actually a detail of what really goes on here on the left without any of the compartments of the cell. This replication, formation of concatenamers, happens by rolling circle replication. So we're looking at this red version here being rolling circle replicated to form concatenamers, and then strand displacement to form the other strand. And RNase 3 is used to process the concatenamers, and these don't have hammerhead ribozymes, so RNase 3 is used to process them, and they end up with these unusual structures in the end. But rolling circle concatenamers and processing by host enzymes. The afsun viroids are slightly different because they have a ribozyme. They enter the host cell, and then the RNA is imported into the chloroplast. It's very unusual. All right, they're copied to make concatenameric RNAs. The concatenamers are processed. They're self-processed by the ribozyme activity of the viroid, and then they can be copied again to make the original strand. They're exported from the chloroplast and moved to the next cell through plasmodesmata. And again, on the right here is a detail of the replication by rolling circle, which is the detail on the right is not shown on the left, and the compartments are not shown on the right just for simplicity. So rolling circle replication, the ribozyme process it into individual genome length pieces. These are ligated into circles by a cell enzyme that would normally work on tRNAs. The ribozyme is an essential part of this. It cleaves the concatenamers. In the previous viroids, the cell enzymes cleave the concatenamers. All right, so where did viroids come from? Well, we think they've been in wild plants for many, many years. We don't know exactly how long, but maybe since the RNA world. Yeah, hung around since the RNA world, and when plants arose, they went into them. Who knows? But they originated in the 20th century by transfer from wild plants that were used to breed modern crops. So today our crops are all monocultures, bred from a single stock. But all the crops we have at some point in the past came from wild plants, of course, and we think that's when we brought the viroids into modern crops. And so now we use globally genetically identical plant breeding lines, which I call monoculture. And so they're all susceptible to these. You can make seeds that are free of viroids by simply looking for viroid-free seeds. And you can plant crops that are free of them, but eventually they will get infected. They're transmitted by pollen from other areas and so forth. Mechanical transmission, contaminated farm machinery, equipment, hands, and plant to plant. And how do they cause disease? This is quite interesting. So these viroids come into the cell, and the cell chops them up using its microRNA machinery. Small RNAs, 21 to 24 nucleotide, are made from their genomes. And we think they guide silencing of host genes. And that is what causes the disease in those viroids that cause disease. They don't all cause disease, of course. Isn't that something? The plant is directing an RNA defense against the viroid, is chopping it up like it would chop up a virus coming into a plant. But in this case, the small RNA silence host genes, and that's what gives you disease. And in fact, you can show that symptom development correlates with the production of these RNAs. They map to this pathogenesis region. So it's from here the microRNAs are produced that silence host genes and cause pathogenesis. All right, that's viroids. All right, satellites. This is a name for a collection of entities that can have either single-stranded RNA, DNA, or circular RNA genomes. And so it's a diverse collection here. But the key here is they depend on a helper virus to be propagated. So they're what we call helper dependent. This word satellite is falling out of favor. Probably better to call them satellite viruses because they are viruses. They get encapsulated, as you will see. And these viruses lack the genes that are needed for replication. And so there are two kinds. They're satellite viruses. These encode structural proteins that encapsulate their own genome. So they make distinct particles from the helper viruses that they need for replication. And then there's satellite RNAs. These are packaged by helper virus proteins, but they also need helper proteins for replication. These sometimes don't encode protein, and sometimes they do. So maybe they're kind of like viroids, except they get packaged in a particle, so they're not really viroids. So here's an example of a helper RNA being packaged in the capsid of a helper virus. So that is, the helper RNA is green here, and that's its capsid, and the satellite RNA is packaged in it. So the satellite RNA make encode proteins, but it needs the capsid to be encapsulated. It also needs the helper to replicate. So you have satellite viruses that have their own capsid proteins and satellite RNAs that are encapsulated by the helper virus. Now, there's a very well-known example, of course, of a helper-dependent virus that we have talked about, and that's adeno-associated virus, AAV. And this virus, which has the funny single-stranded DNA genome with the T's on the ends, well, it's not so funny. It's unusual, I would say. I don't think anyone would laugh at it, right? Maybe a virologist only. Here, they need helper adenoviruses in order to reproduce. Helper adenoviruses provide a variety of helper functions. And in fact, in the last lecture next week, we'll talk about using these viruses as vectors and exactly which adeno genes you need for helper. But these encode their own capsid proteins. So these are bona fide viruses. They just are dependent on adeno for replication. They can't replicate on their own. If you put these in cells, they'll get in and put the genomes in. They're not going to replicate. And that's in some ways why they're good for gene therapy, as we'll see next time. So that's a well-known helper-dependent virus. But we don't call it a satellite, of course. We just call it a virus, not even a satellite virus. It's a virus. It just happens to be dependent on another virus. So we shouldn't belittle it because of that, right? And now we have, go back to the satellite RNAs, the ones that don't encode protein, or at least they don't encode their capsids. There are three classes. Class 1, these are linear, and they encode non-structural protein. And then another class is linear shorter, that doesn't encode any protein. And then we have some circular ones that are short and don't encode protein. And so the way these replicate, the satellite RNA enters the cell, and these are going to be encapsulated, so they'll get in by virus-dependent mechanisms. If they're linear, they're converted to circles, and then they replicate by rolling circle mechanisms by the enzymes provided by the helper virus. These concatenamers are then cut, possibly by ribozymes. We're not sure. Sometimes we're not quite sure the order of things. We think cut and then circularize, or maybe concatenamers cut and circularize. These typically happen in the cytoplasm of the cell. They're not just in plants, but this is about plants. In plants, the satellites cause distinct disease symptoms that are not seen with the helper virus alone. So these all need helper viruses. And the helper viruses on their own do something that cause some kind of disease, or they may. And with the satellite together, they cause something different. Necrosis, which is killing of cells. Chlorosis, which is chlorophyll deficiency. That's what's shown in these bottom leaves here. These leaves are losing their chlorophyll, so they're turning yellow. And they're not defective viruses made from their helper. They're unique sequences. They have no homology with the helper. Where they came from, we don't know. And here's a summary of some of them. There are many of these, but I just want to give you a sense. So here we have the helper-satellite combination. So the helper for adeno-associated virus can be adeno or herpes viruses. And the AAV have single-stranded DNA. Is the particle size, the genome, the capsid proteins. These are vertebrate satellite viruses. And then we have chronic B paralysis virus and the chronic B paralysis virus satellite. Yeah, it's a virus that paralyzes bees, for sure. And the satellite is a single-stranded RNA. This is the particle, genome size, capsid protein. These infect bees. And then we have a plant one, tobacco necrosis virus, with the tobacco necrosis virus satellite. And the satellite, these properties are of the satellite virus. These encode their own proteins. Single-stranded RNA, 17 nanometer particle, 1.2 kb genome, 22 kd capsid protein. Right, so those are satellites. Now, we do have an interesting hybrid which infects, well, for many years we thought they just infected mammals, but you'll see they infect reptiles and birds too. So we have this hepatitis delta virus. It's a mix of a viroid and a satellite. As you'll see, it depends on hepatitis B virus as a helper. And in fact, if you're co-infected with hepatitis delta virus and hepatitis B virus, you have worse liver disease as a consequence. We don't know how that happens. We don't know the reason why, but people who have both are worse off in terms of hepatitis. So we used to call this hepatitis delta satellite, but there's been a movement to not call it a satellite, call it hepatitis delta virus. It's a virus, as you will see. And I think we should throw out satellite entirely, unless they don't encode a capsid protein and they're capsidated by another one. I don't know what to call that. Maybe satellite virus. I don't know. Here's the global distribution of hepatitis delta virus. 18 million people globally are infected, which is 5% of the 350 million people who are infected with Hep B. And here's the prevalence. Asia Pacific mainly. Black is very high, intermediate and low, and no data available for much of the world. It's going down in Europe. It used to be high in Europe, but Asia Pacific is still relatively high. There's a lot of Hep B there as well, of course. You can't have Delta by itself. You need to have Hep B. Delta alone is not able to infect. Well, it can infect, but it won't replicate. So here's the genome of hepatitis delta virus. It is it's a circle of RNA, but it's extensively base paired. So, it's double-stranded there, but if you denature it, it becomes single-stranded. Pox virus is like this for DNA. We call it a double-stranded DNA virus. So if someone asked a question about this, why do we call these single-stranded? It's a good question. This could be a double-stranded RNA, but it's not called that. I don't know why. Good question. All right. Anyway, that's the genome that's in the particle, and this is the antigenome, which actually encodes for the protein. The mRNA is shown at the bottom. So obviously the mRNA is made from the genome, and the antigenome is for replication. This red dot is a self-cleavage site. It's a ribozyme, so this genome has a ribozyme in it. And so I guess these are viroid-like because they have ribozymes and they're circles of RNA, but they're satellite-like because they encode a protein. They encode delta antigen. So there's one mRNA, and it has an open reading frame that encodes for two different proteins, small and large delta antigen. These are copies per average liver cell. It's a 1.7 kb RNA, 300,000 of the genome, 50,000 anti, and 600 copies of the message. So here's the genome again, and the helper virus is hepatitis B virus on the right. Remember, an enveloped virus with a strange gapped double-stranded DNA genome in a capsid. This hepatitis B provides the capsid for delta. So the genome is in the delta particle there, basically the minus strand, base paired, you see it. And then this is enveloped, and in the envelopes are the glycoproteins of Hep B, the large, the medium, and the small versions. And so that's what's provided by Hep B. And then delta antigen, which is encoded in the delta genome, helps package the particles. You see this small delta coats the RNA, then the large is embedded in the membrane and probably attaches it or is involved in packaging. So that's the helper function of Hep B for delta, because otherwise it can't encapsulate itself because delta antigen alone is not making a capsid. And here's a replication scheme of delta genome. Now this is copied by host RNA polymerase II, very much like the viroids that we talked about. So another viroid-like feature. Here's the minus strand RNA that gets into cells. The particles are taken up by the same receptors as hepatitis B virus. And then Paul II starts to copy the negative strand, make a plus strand by kind of rolling circle. And here the red is the ribozyme. So the ribozyme self-cleaves and then makes an mRNA, basically. That encodes for delta antigen. And the red is the ribozyme, the purple is the polyadenylation signal. So the polymerase just keeps copying this, going around the circle and making many, many messenger RNAs. And a subset of those messenger RNAs are not processed and polyadenylated, but they're just ligated to make full-length plus circular RNAs, which are then copied to make more of the genome, the minus strand. And again, the ribozyme cuts the concatemers to form the genome length units that are then ligated together and make the genome that goes into new particles. So that is hepatitis delta virus. Now, up until just last year, we thought that this was a virus specific to humans. We didn't find it anywhere else, but people started looking in genomic data sets and they found hepatitis delta-like viruses in birds and in snakes. They found these by finding the delta antigen coding region, then they found the whole genome. They're quite diverged from the human protein. So here on the right is a phylogenetic tree of delta antigens from human delta virus. That's black. That's genotype 1. And then genotype 2 and genotype 3. So in human populations, human hepatitis delta virus diversifies into 3 genotypes, much like HIV diversifies, but much less extent. And then you have the delta-like antigens from snakes and birds. We call these delta-like viruses because we don't know if they cause hepatitis in the host. We don't know what the helper is. There's no Hep B-like viruses in birds or snakes that harbor these sequences. The thinking is maybe another envelope virus serves as a helper and not hepatitis B. There are hepatitis B-like viruses of birds, but not in the particular birds where they got these from. So it has to be something else. And around the same time these were discovered, other groups found that you could actually package hepatitis delta virus with glycoproteins from other viruses, hepatitis C virus, VSV, dengue, and West Nile. They can all be helpers for hepatitis delta virus. Now, no one's ever seen this in the wild, in nature. That is, no one has seen delta virus encapsulated with one of these glycoproteins, but it can happen. So it may very well be that the snake and bird delta-like viruses are encapsulated by some other virus. So that remains to be seen. Maybe we'll know next year. There's a lot going on in virology besides SARS-CoV-2. Now we have another category called virophages. It's really another helper-dependent virus. They're not called satellites because they were just recently discovered, and the people who discovered them don't want to call them satellites. They find that insulting. I don't need to tell you why, but they think these are bona fide viruses, and I'll explain that in a minute. So virophage. Why is it called a virophage? Well, here we are back to the French. The French like to name viruses, Mimi virus, Mumu virus, and all Mama virus, and all kinds of names, Pandora virus. So this one, the French discovered this. They call them virophages, and the name is derived from bacteriophage. Phagein is Greek for to eat. So this means virus eater. How can you eat a virus? Well, you can't really eat a virus. Well, I guess you can. We eat them all the time. Animals eat them all the time. Birds do. Snakes, everything eats them. But how can a virus eat another virus? Well, obviously it can't. So what happens here is these interfere with their helper virus. And so that's why they call them virus eater. I don't think it's very good, but it's stuck. So these are typically circular, double-stranded DNA virus with icosahedral capsids encoded in the genomes. They will replicate only in cells that are infected with a giant virus. So their helper is a giant virus. And these virophages interfere with helper virus replication. I should just stop calling them virophages. I don't really like the name. They're just viruses that depend on a helper. And many have been found. In fact, you remember the ocean metagenomic data sets that I introduced in our ecology lecture? That's what this is here. Each of these dots is a genomic data set. And you can mine them for these virophages. So they're all over the place. They're in oceans all over the world. But these are some specific isolates. Here's Sputnik. That was the first one. Again, the French discovered it. They called it Sputnik, and they called it a virophage. It's from a cooling tower in Paris, where they originally got Mimi virus, which infects amoeba, and they found with it this Sputnik, which has this size genome, and has a lot of proteins. So these are bona fide viruses with lots of open reading frames, respectably sized genomes. Although I think a 300 base genome of a viroid is pretty respectable, that it can work with such a small thing. Here, another one, Ma virus, from Cafeteria rhombogensis virus, and then a whole bunch of others. Here's one. Again, the French. I'm going to point out the French sense of humor, and I don't mean to insult them at all. I think it's funny. This one's Sputnik 2. This was isolated from the contact lens fluid of a patient in France with keratitis, and they called it lentille virus, which means lens, right? Jokesters. I think that's pretty funny. And there's a bunch from Yellowstone Lake. We're not sure of the hosts there, even in Antarctica. All right, so there are lots of virophages out there. And actually, this story gets more interesting each year. And next year's course will be even more interesting because a lot of people are a number of people are working on these. Okay, so here's the capsid of Sputnik, solved three-dimensional structure. It's quite large and respectable. And here's some interesting electron micrographs to show what these do. Now, if you infect a cell with mimivirus are these first discovered giant viruses on the left here. You can see them. Icosahedral capsids with the fibers sticking out. And they found in cells infected with mimivirus this Sputnik, which are these smaller particles shown by the arrows. All right, and the Sputnik depresses the yield of mimivirus from cells, so it inhibits it. So it needs mimivirus as a helper. Sputnik will not replicate without mimivirus. Yet, it's like biting the hand that feeds you. It interferes with the yield. And it makes weird mimivirus particles. So the normal particle is shown here in panel A, and then look at the bottom in F. There's a screwed up particle where it's not even completely encased. That's caused by Sputnik. On the top, panel B, you can see this particle, this mimivirus particle is empty, but it's got some Sputniks in it, right? And look at this one. This looks like those pastries with all the layers. That's a mimivirus where something's gone wrong, right? And it's just accumulating this protein on the outside. In the bottom, same idea, wrapped around and around. Anyway, so that's what happens in a cell co-infected with Sputnik and mimivirus, and how it does this, we don't know. But it interferes with the yield. These virophages have interesting potential activities. So, by the way, if you're interested in this depression of yield of virus, when you infect a mimivirus-infected amoeba with Sputnik, the yield of the mimivirus goes down 70%. And here are some other virophages. Ma virus is a virophage of a giant virus that infects this guy here, Cafeteria rombergensis. This is a marine phagotropic flagellate. Phago means it eats. It phagocytoses everything. That's why it's called cafeteria. It will eat everything in the ocean. Well, it has to be small, of course, but it's a robust eater. Anyway, this is a virophage of a giant virus of this organism here. There's also this organic lake virophage of phycodinoviruses that infect algae. Now, the people who have studied this organic lake virophage, they believe that this virophage can influence the composition of the microbial community in a lake. And so they have tested this by modeling the effect of virophages on a population of microbes that's infected with a lytic virus. So the virus would kill the microbes. Without the virophage, you have cycles of virus reproduction, cell death, and cell growth or blooms, algal blooms, right? If you add the organic lake virophage to this equation, you get more blooms. The frequency of blooms increases. And so it's suggested that the virophage reduces the mortality of the host alga, which is caused by the phycodinovirus that infects them. And, this is an Antarctic lake, organic lake. So the authors of that study suggest that these lakes have long cycles of daylight and darkness, and a decrease in cell killing by the virophage may be essential for maintaining the stability of the microbial food web, because there's not a lot of light for these cells to grow, for these algae to grow. They need light to grow. And so maybe the virophages are protecting them and maintaining the ecosystem. So this is another example of, microbial viral ecology. And so impact on ocean ecology, we think they also exchange genes. If you sequence the genomes of these virophages, you see genes taken from the host and the giant virus helper. All right, so the last thing here is, are they satellites? So the people who discovered these, when you go to meetings, they have vigorous arguments about why we shouldn't call them satellites. So they're just called virophages or helper-dependent viruses. And here's the argument. Like many autonomous viruses, they depend on transcriptional machinery, right? So many small DNA viruses, depend on Pol II from the host, right? We don't call them helper dependent. We just call them viruses. In this case, these satellites or virophages, they just need transcriptional machinery from another virus, not the host cell. So why should we call them even helper dependent, right? You get my point? And I think it's a good point. We should just call them viruses. They happen to depend on both the host and another virus. Now we leave the realm of viruses to infectious proteins. Prions, infectious proteins with no nucleic acid. And look, someone lives on Prion Road. How cool would that be? That's no throughput. Even cooler. Anyway, these get in the news from time to time. Bovine spongiform encephalopathy or mad cow disease, Creutzfeldt-Jakob, Scrapie, Kuru, chronic wasting disease of deer and elk. And Stanley Prusner studied these for many years and got the Nobel Prize for understanding how they work in 1997. Here's a story. If you know George Balanchine, he was a dancer and the head of the New York City Ballet for many years. He died of Creutzfeldt-Jakob, and his cells were examined at Columbia, and they found that he had some degenerative neurological disorder, which was Creutzfeldt-Jakob. That's what he died of. Interesting article here. Okay, so what is this? This disease is a transmissible spongiform encephalopathy. And the encephalopathy is a disease of the brain. These are fatal neurodegenerative disorders of mammals, and not just humans. Others, as you'll see. There are thousands of human cases every year. About 1% of those arise by infection. The rest are sporadic. And by 2002, as you will see, 120 humans had contracted Creutzfeldt-Jakob, which is one of these TSEs, from eating meat from animals with mad cow disease, or BSE. It's pretty scary. You might not, after this lecture, you might not eat meat again. If you do, you can't cook it. It doesn't kill it. No, it doesn't. Cooking doesn't kill it. Even if you make your hamburgers well done, could still have prions in it. I mean, it's pretty rare, but transmissible spongiform encephalopathies, they happen to animals. There's bovine, which is also called mad cow. There's chronic wasting disease of deer, elk, and moose. There's exotic ungulate encephalopathy. That's an ungulate there. There's feline spongiform, both domestic and the great cats. There's Scrapie in sheep and goats, and there's in minks, transmissible mink encephalopathy. So a lot of them. Then there's the humans, Creutzfeldt-Jakob, which we'll talk about mainly here. Fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, we'll talk about Kuru, and variant Creutzfeldt-Jakob disease. These are all human TSEs. Transmissible spongiform encephalopathies. So we have explained encephalopathy. Transmissible, I don't have to explain to you. Spongiform means your infected brain has sponge-like holes throughout it. Here's a section of a brain of a person with a TSE. These holes should not be there, right? It should be all smooth. This is typical. And these individuals have a lot of issues. They have severe psychomotor dysfunction. Each disease has a characteristic symptomatology. The diseases I just showed you here, each is characteristic. And the symptoms depend on what part of the brain is damaged, because these prions can go in different places. So this all starts with Scrapie, the first TSE that was recognized. These are sick sheep, and you can see the hair is missing. Anyway, whatever it is, they rub themselves on fences. That's why the disease is called Scrapie, and it rubs off their fur or hair. And these animals get motor disturbances, uncontrolled trembling. They shiver uncontrollably, tremblement du mouton, paralysis, weight loss, and they die. As soon as with the onset, they're gone in 4 to 6 weeks. You can't stop it. There's no cure, no prevention. And this has been recognized as a disease of European sheep for over 250 years. You can see it in the literature. It is endemic in some countries. In the UK, 1% of sheep every year come down with Scrapie. All right, so this is a problem because we feed sheep to cows. Keep that in mind. Why do we feed sheep to cows? So they grow faster. Protein as opposed to grass or whatever, corn. Farmers were the first to figure out that their animals could transmit Scrapie to healthy ones. They said, it's an infectious agent. 1939, people started studying this. They found infectivity from sheep brains could pass through filters. So you can pass it from sheep to sheep after filtering it. So that said, well, it must be a virus. Turns out to be highly resistant to UV ionizing radiation, formaldehyde, autoclaving. If you autoclave this stuff, it's still infectious. So eventually the idea came that there was no nucleic acid in it, and it was clearly not a typical infectious agent. And, Prion worked on this for years, and people didn't believe him. At meetings, people would say, you're out of your mind. This is a virus, and there's a little bit of nucleic acid, and your experiments are all wrong. Well, he turned out to be right. Great story. You should read his autobiography. It's amazing. Anyway, here's one of the early experiments. This is the inactivation of virus infectivity by ionizing radiation. And here there are a whole bunch of viruses where we're looking at the molecular weight of the nucleic acid, the kilobases here on the right, and on the bottom, the dose in rads. So more radiation from left to right. And, the DNA viruses are all on their own curve, and the big genomes don't need as much radiation because they're big targets. It's easy to hit them. And you get down to the small ones like SV40 and Prion, you need more radiation to hit them. And here are the RNA viruses as well. Same trend, large to small. But look at Scrapie. The Scrapie agent is all the way down here, the highest amount of radiation needed, apparently a very, very small target, which suggests it's not a nucleic acid. So that was the first bit of information that said, there's something different here. And Prion took this and ran with it. So it turned out that studying animal and human TSEs, which came up later after Scrapie, people said, wow, this same histochemical abnormality. So people had studied Scrapies for years, and they'd looked in their brains, and they had ideas of what was going on. It was all published. And then suddenly people started studying human TSEs, and an astute person said, the brains look similar. Defects in plasma membrane formation, vacuolization of neuronal cells, loss of neurons, making that spongiform appearance, accumulation of GFAP, amyloidosis, deposition of amyloid, and fibrils, which we know are made by misfolded prion protein, which ends up being the key here, which I'll tell you about in a moment. And eventually people started studying human TSEs. They would make brain homogenates, and they would inject them into various species, like mice and hamsters would get disease, only it took a long time. And these animals, after many months or years, end up getting ataxia, dementia, and died. Eventually they could figure out how to speed up the assay. But after you inoculate this agent, whatever it is, it accumulates in lympho-reticular secretory organs, spreads to the CNS, and there you get astrocytosis, vacuolization, and loss of neurons. No inflammation. There's no inflammatory response, no cytokines. There's no antibody. There's no cellular response. So whatever you're injecting into these animals from another animal, it's not causing an immune response. These TSEs are insidious because in humans and animals, you don't know they're there until symptoms develop. Now we do have a diagnostic test, but it isn't as widely used as it should be. They're untreatable. Once you have these symptoms, there's no way to alleviate them. 100% fatality. And that's still the case today. When someone develops a TSE, they're going to die. There's nothing you can do the same. Then we don't have any drugs to prevent dying. So in 1967, a mathematician, Griffith, said these are proteins. All this information about the inactivation, it says they're a protein. Of course, people thought he was crazy, and Prion took this up, and they thought he was crazy, but he turned out to be right. In 1981, he identified infectious protein complexes in the brain of Scrapie sheep. He purified the protein. He transmitted it to animals. He called this a prion, a proteinaceous infectious particle. He moved the letters around, obviously, right? Prion. Prion. He originally came up with Prion, but he didn't like Prion, so he made it Prion. That's a good word. I like it a lot. And the prion is encoded by a gene called PRNP, and this gene is essential for the pathogenesis of TSEs. The PRNP gene is a cellular gene, and its protein is causing this disease. How does that work? The pathogenic prion is a conformational isoform of a normal host protein, which we call PRPC, prion protein cellular, So this is a normal cell protein found on the outer surface of neurons. It's GPI anchored, but it occurs in an abnormal conformer called PRPSC. C stands for Scrapie, kind of in honor of the sheep, because all this initial work was done with sheep. So the abnormal conformer, which you can get from sheep brains or human brains, if you introduce that into another organism, it causes the normal protein, PRPC, to convert to the pathogenic conformation. Right? Normal conformation, PRPC, abnormal PRPSC, that causes disease. PRPC doesn't cause any disease. So here's the story. Early on, Prusiner found that you could distinguish PRPC and PRPSC by a difference in digestion with an enzyme called protease K. You would just take the proteins, extracts of brains of infected animals, you could treat them with proteinase K. The C, PRPC, which is shown here, is completely digested by protease K. But the PRPSC is just cut to give this 27 to 30 kilodalton fragment. So it cuts the N-terminus off. So he could use this as an assay in his experiments. All right, so what happens here? We have PRPC, and we now know the structure of PRPC. It's converted in some way to a conformationally altered protein, which has a lot of beta strands and beta sheets, and this is the pathogenic protein. And recently, the structure of the PRPSC was solved, and it's a very extended beta sheet structure, as you can see here, which is kind of illustrated here. So it goes from alpha helical mainly to mostly beta strand. All right, so that's what's going on here. All right, so the Prion hypothesis was finally proven by this experiment that if you take mice and you knock out both copies of the PRNP gene, you cannot give them a TSC by infecting them. So you could take mice with TSCs and make extracts of their brains and inject them into other mice. They will get a TSC. But if the mice are knocked out for PRNP, they're resistant. So that's the key. PRNP is making PRPC. When you introduce PRPSC, it's converting it. How do you get this PRPSC? You can get it by infection. You'll see you can eat it, or you can get it by transfusions and so forth. Or sometimes there are rare mutations in the gene that cause the protein to misfold spontaneously from PRPC to SC. And so either by eating it or getting it or spontaneous mutations, it accumulates, the pathogenic form accumulates in the brain, and eventually you get the symptoms of these TSCs. And there are three types of spongiform encephalopathies. There's infectious, familial, and sporadic. So infectious or transmissible, TSC, you have the SC form and it's transmitted to another organism by eating or some other method, which you'll see in a moment. It converts your PRPSC into PRPSC, which makes these long beta sheets, and that's pathogenic. That's the infectious form. Then we have the familial or genetic form, where you inherit a mutation from your parents in the PRNP gene, and that causes spontaneous conversion of PRPC to SC, which then converts the remainder of your PRPC into sheets. Familial. And then sporadic, down here, you have a normal gene and you don't get infected, but just your protein misfolds. And once you have enough of the misfolded, it'll convert everything else, all your other C, into the sheets. All three of these diseases can be transmitted experimentally to animals by inoculating them with infectious material or by feeding them infectious material. All right. So you could give a mouse an infectious dose and then take the brains out and infect another animal with that. You can take an animal with a mutation in PRNP that develops TSC and you take that extract of the brain, you can infect another animal. Or you could do the same thing with sporadic. So in the end, PRPSC is infectious. It's an infectious protein. Now, human TSCs, there are all three kinds: infectious, genetic, and sporadic. So this is genetic or familial, but I can't say familial easily, so I say genetic. In fact, let's look at the infectious or the transmissible one. This first example we learned was Kuru. We'll talk about this. Or spread of the disease by transplanting infected corneas, hormones, or transfusion from patients with CJD, another human TSC. So we learned early on. So, you can give corneal transplants. Some of these people got TSCs because the donors had it. Hormones you can purify from people. They have TSC. They have prions in them. They can give you TSC. Transfusions, all from CJ patients. You didn't know they had CJ. It was probably too early. They hadn't developed symptoms yet. And so this started an outbreak of CJ. So this is infectious, obviously. We call it iatrogenic because it's associated with a medical procedure. Bovine spongiform encephalopathy is another human TSC we get from eating beef. And it started because we fed infected animals to cows. I'll tell you about that separately. And that led to variant CJ, and a new human disease you get from eating BSE beef. So the BSE is a cow disease. And then the human disease we get from eating the infected cows is called variant CJ. All right, let's start with Kuru. Kuru is the name for a TSC found in the four people of New Guinea. Here's New Guinea, okay? There's Australia down there. This is a disease with a 30-year incubation period. It was originally studied by Carlton Gajdusek, American virologist, and he noticed that this disease was spread mainly among women and children. Why? Because when someone died, they would eat the brains of the dead person, ritual cannibalism. Only the women and children were allowed to eat the brain, and so the men didn't. They didn't get this disease. So he told them they shouldn't do this, and he convinced them to stop, and Kuru ended, and we don't have it anymore. But that's a TSC. These people are dying of sporadic or maybe familial TSC. You eat the brain and you get infected. We have sporadic CJ, where you just have spontaneous misfolding. There's no genetic basis, no infectious basis. 1 in 2 per million people every year in humans, typically 50 to 70 years of age. This accounts for most TSCs. It appears with no warning, no epidemiological indications. This would mean like eating BSE beef or something like that. They have normal PRNP genes, and they can transmit it to others. If you get their corneas or their blood or their hormones or whatever, you can transmit it. And we think Kuru was established probably in New Guinea by eating the brain of a person with sporadic CJ. So you only needed one, and the women and children ate that brain, and they got it, and then people ate their brains, et cetera, et cetera. It only had to happen once, right? And then finally, familial. It's an inherited disease. There are autosomal dominant mutations in the PRNP gene that predispose them to misfolding. And these people, before they have symptoms, they can donate organs, corneas, blood products. They can be infectious. They have transmitted CJ to others. So here are the worldwide CJ deaths. Creutzfeldt-Jakob can be, it's mostly sporadic, but it can be transmitted by transplantation. And from 1979 to 2017, these are the number of deaths in the U.S., 200, now up to 500, population increase mainly, and this is deaths per million. Age-adjusted death rates is more or less stable, maybe slightly going up here. So 2.5 deaths per million people. This is not, it's rare, but it happens, clearly. All right, so that's Creutzfeldt-Jakob. Now let's talk about BSE, bovine spongiform encephalopathy. So at some point in the UK, cows started developing TSCs, which was called bovine spongiform encephalopathy, or mad cow disease. And I'm going to tell you why this is a form of cannibalism. It resulted from feeding cows processed animal byproducts, including sheep with Scrapie. They just throw all the animals in a vat, and process them. There's protein supplements, so they grow faster. In the 1970s, they changed the way of preparing this, what's called meat and bone meal feed. They changed it in a way to save money, and this allowed now the Scrapie proteins to pass from the sheep into cows. And so people would throw the dead Scrapie into the vat. Sheep, they didn't care, they didn't know better. And anyway, the processing method worked until this change happened. And so this transmitted the disease to cows. They stopped feeding cows other animals, and the outbreak stopped. And it's quite clear that human consumption of BSE-infected beef transmits bovine TSC to humans, and this results in a new disease called variant CJD. It has characteristics of many kinds that are different from human CJ, so that's why it's called variant. And so here's the epidemiological curves. The estimate is that 1 to 2 million cattle were infected with Scrapie prions. And the problem here is that it got into the food chain, right? Because the incubation time is about 5 years, yet cattle come to size in about 2 or 3 years, then they're slaughtered for meat. So they could have the prions in them and you wouldn't know it. That's why it got into the food chain. But when the outbreak in cows began in the UK, so here we have thousands of cases in cows, here in orange, BSC, started going up and up. And in the UK, they said, oh, this is not good. This could get into people. So they stopped feeding the cows, the sheep, and then eventually that went down. But then a number of years later, because the incubation period in human, we had variant Creutzfeldt-Jakob. The blue are variant CJ in people, and that went down eventually when, lag after the cow went down and no more meat, no more tainted meat in the food supply. Some of these people who had variant CJ donated blood. They didn't know they had variant CJ, and we had a couple of cases of variant CJ transmitted by blood transfusions, the green there. So this is really an unfortunate situation. The cows got sick and they spread it to humans because of consumption of cow meat, of course. Now, what's going on with this? We monitor BSE in the cattle, of course, and we see new cases all the time, which are probably sporadic. They're not from eating meat of other animals. They just happen from sporadic misfolding of the protein in cows. And so the key here is, of course, to protect the food supply, but we only check, at least in the U.S. and Canada, less than 2% of slaughtered cattle are tested for BSE. Now, diagnostic tests have been developed. I don't know how much screening is done anyway. And we're looking for drugs that block these, but we haven't found any to help in case anyone gets infected. And here are the numbers. They're not big. And whenever you see one, there'll be a single case. You see many years there's a single case. These are BSE cases in North America, in the U.S. and Canada, 26 since 1993. Not a lot, but whenever there is one, everyone freaks out because who knows how many others there are. And the reason they pick it up is because they'll have a cow getting older for whatever reason, milk production or something else, and it develops the symptoms, and they say, oh my gosh, how many other cows have it? So this is a bit worrisome and makes some people not eat meat. And I don't have time to tell you these stories, but if you want to read about them, I wrote 2 stories of very interesting issues. First, a case of prion disease from contaminated beef. This is a man who lived in the UK for a few years and then went and lived elsewhere, somewhere in the Middle East. And then 15 years later, developed a TSC and died. And it turned out he had eaten burgers in the UK at a time when there was BSE. So if you lived in the UK during that outbreak, you cannot give blood. You are forbidden to give blood, even if you never touched any beef in your life. And then another very interesting story, this is in the emergency room. An individual was acting really strangely. He shot himself. I think it was in the emergency room or before, but anyway, he came into the emergency leaking brains essentially, and they found later that he had a prion infection, so they destroy all this incredibly expensive equipment because you can't decontaminate it. There's no way to decontaminate these prions. They're so resistant. We have developed an interesting assay where you can do nasopharyngeal swabs. This is very interesting that this is a way you can diagnose SARS-CoV-2. You put a brush way up into the olfactory area, in this case sampling the neuroepithelium because the prions are moving from the brain. You could do this with people or cows, and then you subject it to a kind of PCR for proteins. You add your sample, which might, if it contains, beta sheets of the PRPSC, it will cause the formation, and you add a little bit of normal protein, it will cause the formation of longer sheets on incubation. Then you break these up by sonication, and you repeat the cycle, and eventually you get lots and lots of protein. You do like 20, 30 cycles of this. You get lots of the sheets. So you can do an assay for this in cows and people now, so that should help. So let me summarize these 3 epidemics of Creutzfeldt-Jakob. These are the number of infected patients each year from 83 to 2009. So on the top, this is our iatrogenic CJ caused by injection of growth hormones purified from humans. And of course, they had CJ. So this is an outbreak. We eventually recognized what was going on, so we stopped this practice. And now we use recombinant growth hormones, of course. And then we have duromotor grafts, people getting grafts from CJ infected people. And then, of course, the variant CJ from eating prion contaminated beef. 3 overlapping outbreaks. Look at that. There is a species barrier of prions I want to tell you about. If you inoculate diseased material into the same species, you get disease. If you go in a different species, it's inefficient. But if you match this PRPSC and the C, then you get efficient production. So the sequence of the protein is a barrier to species transmission. So here's an example. If you inoculate hamster PRPSC into a mouse, you don't get disease. But if you inoculate hamster PRPSC into a mouse that you've made transgenic for hamster PRNP, then you get disease. So they have to match. The problem is that bovine PRPSC has a broad host range and can infect mice and many other animals, including humans. So we eat the meat of a cow and we get it because it has a broad host range. So some prions can overcome the influence of the primary sequence, and that's why BSC is a concern. Another concern that has arisen since 1967, chronic wasting disease of deer, elk, and moose. And this is a disease that causes a TSC in these animals. It's been reported in the U.S., 26 states in the U.S., 3 provinces of Canada, South Korea, Finland, Norway, and Sweden. And it's either in captive herds, where it can be up to 90% of mule deer and 60% of the elk can be positive. In wild deer or free-ranging, as high as 15% or more. Many thousands of infected deer and elk have been identified since that first animal. So why is this a concern? Well, we're afraid it's going to go from these animals to someone else. And it's been shown that grass plants can take up, retain, and transport infectious prions from the soil. So you can add prions to the soil. They'll get into the roots. They'll get taken up into the leaves. Or you can add the prions to the surface and then grind up the plant. And you can feed it to animals and they'll get infected. So we're worried that these deer with TSCs they can urinate on the plants and contaminate them. And maybe they would infect other animals as well. So if you take cervid PRPSC and inoculate into mice, you don't get disease unless the mouse is transgenic for cervid. So there is a host restriction here, which is good. If you take cervid and put into mice transgenic for human PRNP, there's no disease. So this would suggest there's no threat to humans, right? But again, it is an animal model, so we don't know if that's actually correct. If you take deer PRPSC and put it IC into cows, you get disease. However, if you feed the cows, they don't get it. So maybe, cows are not going to get inoculated IC. So maybe this is a good thing. But we're worried that, prions in pastures put down by deer could contaminate and transmit to cattle. There's no evidence for this so far, but we're worried about it. And the problem here is that the incidence of CWD in cervids is increasing. So it's estimated that between 7,000 and 15,000 infected animals like deer are eaten by humans every year. People like to eat deer. So far, we've not seen any transmission of CWD to humans. So we think the risk of human infection with CWD prions is low. However, we tell hunters to be careful. There's a whole site devoted to this. If the animal is acting weird, leave it alone. When you dress an animal, wear gloves. Don't saw through the bone. Don't cut through the brain or the spinal cord. Minimize handling of those tissues. Wash your instruments thoroughly. Don't eat brain, spinal cord, eyes, spleen, tonsils, lymph nodes where we know the prions accumulate. And don't eat meat from animals that test positive. Be careful, hunters. So finally, let's just talk about the history of these diseases. It's likely that all of this started with some naturally occurring disease in sheep as far back as 1720. So the blue boxes are naturally occurring diseases. And we think maybe these sheep sporadically developed Scrapie. They may have transmitted it to deer and elk in the 60s, or maybe the deer and elk developed their own sporadic disease. And then from the sheep, it went into mink. It went to cows, as I've shown you. From cows, it went to other cows. We feed cows to other animals, lions and tigers and so forth. We've transmitted it to them. We feed cow meat to cats. Cats have got, we think maybe feline spongiform encephalopathies came from cows originally because they are fed cow meat. Of course, we got variant CJ from eating cow meat, and we have transmitted via blood products CJ from humans with CJ. And it's possible that some human CJ arose, separately, because not all came from the BSE epidemic. Some arose separately from sporadic humans, and their products then enter the iatrogenic chain. So it's quite an interesting situation, and we don't, as I said, it's uniformly fatal. We have no drugs yet to prevent it, and people are still working on this. Next time, we will end this course by talking about therapeutic viruses, how you can take a virus and use it for good.